This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1313, recorded on April 10th, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
It's good to be here, and it is a lovely spring day. It's a little breezy, but it's 67 Fahrenheit, 19 C, and just nice, clear blue skies. Love it.
Yeah, I got up this morning. It was just above freezing. I had to wear gloves and a cap.
Sure.
Now it's 18 C and sunny here, which seems like pretty good, but it's going down already.
Yeah, it's spring. It's not summer yet.
It's going to be 5 overnight. Next week, the overnights are going to be 15, 16, right?
Yeah.
So we're starting to get a little better, and we got
getting
rain. Also joining us from Austin, Texas, Rich Condit.
Howdy, everybody. We've got, I think we did several weeks ago, fake spring.
Yes, right.
And then we did fake summer, punctuated with some fake fall. And I think we may have real spring now. And I'm dreading the onset of real summer. 77 degrees, overcast, scattered showers. And we're going to get scattered this and that through the next several days, which is fine with me. Rain works here for me in Texas.
We could actually use some here in the Northeast. It's been unusually dry.
We had rain this week, I think.
Yeah, they're a little bit off and on, but it's we're way behind schedule.
I was in Kansas last week. It was really dry there.
Oh, how was that?
It was very nice, actually. I had a lovely visit.
Good.
I met with a bunch of faculty and students. They were all enthusiastic. And, I hate to say this, but sometimes you go to like top-notch institutions and people are kind of
ho-hum.
Yeah.
Ho-hum and laconic. But, this is not a top-notch. It's good, but it's not, if you look at R1s, it's not in the top. But yet everybody's enthusiastic. They're doing interesting stuff. I met with some students. We did a podcast, which was really good. And this is what tells you. So I record it and David Renata edits it. And usually he has to zoom in on the speaker because everyone else is not paying any attention. And he said everybody was engaged totally. So he did the 5-person view for the whole thing.
Cool.
Isn't that great?
That's
They're looking at the speaker and they're nodding, instead of just looking around.
When's it my turn? Yeah.
I mean,
let's redefine top notch, okay?
Yeah, yes.
So I'm just thinking about R1s because they mentioned that they have these NIH grants, which are meant for institutions that are on the lower half of R1 funding or something like that. So I'm not trying to denigrate it. Everybody can't be in the top 10 percentile of R1
That's
grants,
not how
right?
math works. Yeah.
No, that's right. But, they're all enthusiastic. They go every day. They do their work. They publish. And I talk to them and they have, good things. I really enjoyed it. Kansas and, Kansas City, there are 2 Kansas cities. One is in Missouri. That's the big one. That's where you
Right.
fly into. And that airport, they don't use TSA. They have their own private contractors.
How do they manage that?
The state pays for it.
Yeah, that I discovered during this whole fiasco that is, I don't know if it's not uncommon, but it's a thing.
And that's Missouri. And then you drive to Kansas. But in Kansas, there's another Kansas City, but it's much smaller. And so the big airport is MCI, which is not even a Kansas City.
I'm restraining myself from bursting into song here.
But, they're called the Jayhawks because that was they were fighting Missouri in the Civil War, right? When Missouri was a slave state and they wanted Kansas to be a slave state. And Kansas said, we would like to be a free state, please. And they fought and they were called the Jayhawks, a combination of blue jay and hawk because they are fierce.
Cool.
I like that. And there are also 2 viruses, at least 2 are named after Kansas, right? Bourbon virus and Heartland virus.
Heartland, yeah.
The 2 2 counties named after that or named that way. So yeah, I enjoyed it very much. It's smack dab in the middle of the USA. Okay? It's one of those rectangle states, and it's right in the middle.
This was Kansas State?
No, University of Kansas.
University of Kansas.
So
Kansas State is
in Manhattan.
Right, and that's where Lorena Passarelli was. Is that right?
Yeah, they retired. Both of them retired.
Yeah, they've been traveling all over the place. All
And
right.
so one guy came from Kansas State. It's about an hour and a half away. Jürgen Richt.
Oh, yes, I am
Interesting
familiar with
fella.
Jürgen Richt.
He's very, and you should watch the TWiV because he was very animated. I wanted to ask him a lot of things, but we mostly talked about move flu, as he calls it. And they had these. So he's part of some center, CRAP or some. I can't remember the initials, and they had these cool jackets with it, and I admired them. And I just got one in the mail today.
Oh, that's cool.
He sent me one. Swag. And also, yeah, here it is. CZID. You can see it on the pen.
CZID. I was going to say, I hope it's not CRAP. Because that
Center for Emerging and Zoonotic Infectious Diseases. It's cool. I love getting pens. Here's another one, a blue one he gave me, and he gave me this nice jacket. I posted a picture on Instagram, so check it out. Anyway, if you like these programs, we would love your support. We talk about science. We do papers, but we also talk about science in the news because it's important for you to know. You can go over to microbe.tv/contribute. There are a number of ways you can help us out. The Rosenfeld lab has moved to Philadelphia. They're looking for a postdoc and a technician to work on enterovirus pathogenesis and immunity. They're interested in understanding the effects of cross-reactive antibody responses. They want to make animal models, mouse models, and they want to develop new vaccine platforms. So give her an email, rosenfelda@mlhs.org. ASV, American Society for Virology. The 2026 meeting will be July 27th through 30th in Minneapolis at the University of Minnesota. You can go to asv.org and you can learn more about it. Accommodations, a scientific program, biographies of the 3 people on the local host team, Yu Ying Lian, Lou Mansky, and Wei Zhang. Lou Mansky just got some award. What did he get? Oh, I forgot. Congratulations, Lou. I know he listens.
Whatever you just got, congratulations.
Whatever you got. You can also learn about Kid Care, right? There's a deadline for Kitty Corp. May 22nd. And if you get to those links, you go to the top menu bar where you'll see ASV 2026, and there's a drop down. Early bird registration is open till June 1st. Okay, so a couple of news items. This one just popped up yesterday. Very interesting. It's in KFF Health News. U.S. scientists sequence 1,000 genomes from measles, a disease long eliminated with vaccines. So, there's been complaining that only 8 genomes have been released so far from the U.S. epidemic. And why do we want genomes? To see where this is coming from. And also you can see if it goes from Texas to North Carolina to Utah or whatever. And so it's circulating in the U.S., which means we have endemic circulations, which means our certification of elimination is gone. And that's why they're restricting it release until now. They can't wait anymore. So they released 1,000 genomes and there's more to come. And
weren't people like throwing fits about China not releasing enough genome data early in the pandemic?
I mean, yes, totally. Yes. But they say it's because they don't have enough people, because a lot of people got fired.
Well, right, because they fired half the CDC, but that's.
Anyway, Christian Anderson is quoted as saying, we should see a report in April, assuming no interference, no political interference, because you can do the analysis pretty quickly to see if it's, whether it's continuous spread, and that's what we want to know. And we also will see if other mutations are arising of interest and so forth. This shouldn't what they say here is a number of people in this article that were even talking about this is nuts. Someone said, and I applaud the science, but the more urgent need is to get measles under control as quickly as possible, right?
Yeah.
Get people vaccinated.
There shouldn't be 1,000 measles genomes to sequence
No.
No.
in a season.
No. Oh, well, anyway, that's...
I do think the
nice
science
article,
is interesting. The notion that you can tell whether there are isolated infections or spread.
Yeah.
And that's a critical piece of information.
Absolutely.
So this is a good article. Here's a quote, although the exacting director of CDC, Jay Bhattacharya, says vaccines are the best way to prevent measles, he has undermined vaccine policy. Two-faced.
Yep.
Speaking of which.
Yes. You mean the next story?
Yeah.
Yes.
Yeah, whose is that? Who put it in?
I put it in.
Go ahead. Go for it.
This is an article from the New York Times. Says that top CDC official delays report on COVID shots effectiveness. That official is none other than Jay Bhattacharya. And this is a report that was supposed to be published in MMWR, one of our favorite journals, Morbidity and Mortality report, weekly report from the CDC. And it just describes the effectiveness of the COVID vaccine, which of course is effective. And Bhattacharya has, I guess unilaterally, basically, said that he's going to delay its publication because he wants to review the methods and see if the statistical methods applied are appropriate for the study, blah, blah, blah. Okay? And this is nuts.
Yep.
It's the same statistical method that has been used all along. Of course, I suppose he could argue, if he wanted to, that all along those statistical methods have been inappropriate, and so vaccines don't work at all.
From his deep understanding of statistics, yeah.
Right.
Yes, as a health economist, right?
Right.
Right. So, at any rate, this is just, in my mind, an example of the hierarchy in the CDC, at least, tinkering with the facts.
And this is
and trying to promote their own ideology or perspective on what this is all about.
And to be completely clear, this is a totally wacky thing to happen for a CDC director, acting or otherwise, to stop a specific paper from appearing in MMWR. That is not something that the director of the CDC would normally be involved with at all. This is a journal. They've got their own peer review process, their own editorial team. They're on top of this. But yeah, this is just political shenanigans.
So Andrew Nixon, who is just ignorant of everything, he's the spokesperson for HHS. He said it's routine for CDC leadership to review and flag concerns. It's not.
No, not in MMWR, no.
And he says Dr. Bhattacharya expressed concerns about the observational method used in this study. Mr. Nixon, do you know what an observational study is even? Bhattacharya, do you know what an observational study is? Ay.
And
So-
I would like to mention the article, the New York Times article is written by Apurva Mandavili, who I worked with as an editor at Nature
cool.
Medicine for a number of years.
Really? She was
Yeah.
at Nature Medicine?
Yeah.
Really? I didn't know that. That's cool.
So on Monday, I think it was, I had the privilege of giving a lecture to students at the perimeter campus of Georgia State University via Zoom, invited by one Jeffrey Marr, on vaccines, which I divided up into history, science, and public health. The history being my favorite, which is the smallpox story. The science being the fundamentals of viruses and how vaccines are made, basically my YouTube video that I posted a while ago. And the public health, talking about what the roles of the states and CDC are and what the recent changes have been, and then highlighting just one example of the changes being the hepatitis B virus, get rid of the birth dose in some circumstances, and what the likely what the history of that vaccination is and what the likely impact of that change will be. At the end of the talk, the host asked me whether I trusted CDC anymore. It's a really good question. And my answer was no.
Yeah.
I mean, that's a qualified no, because I still go there to look at the information if I want some background information on this or that. But I can go there with knowing some fundamentals about what the truth is. So if I see BS, I can recognize it, but not everybody can do that.
Yeah.
And this is a perfect example of the CDC trustworthiness being compromised by this. And then, and it feeds into the larger conspiracy of people who think that science has always been corrupt.
Yeah.
And that is an even bigger tragedy.
Yeah. I think I trust a lot of the scientists who are still there, but not the
Absolutely.
leadership.
Yeah.
The
Absolutely.
leadership, nope.
Yeah, and the leadership is a filter through which all this other stuff has
Yeah.
to go.
Yeah, I trust the scientists who are still left.
Yeah.
I trust the ones who are gone from there even more when they speak out publicly. But yeah, the leadership at the top is just.
But for example, the measles genome sequencing, it wasn't even done at the CDC.
Right.
It was done at the
Right.
Broad Institute in Boston, right? So I don't even know what the hang-up was. All they had to do was send them the samples. It could have been done a long time ago. So there's something going on in there. But you understand that the goal of this administration is to destroy the CDC and make everything done at the state level.
Right.
They want to have 50 state health departments recommending stuff.
Well, they want to
states,
not have any health departments at all is kind of the direction.
You think not?
Yeah,
Really?
I mean, this notion of devolving things to the state is a way of saying we shouldn't have it, basically, because everybody knows that the states do not have the capacity to do what the CDC does. I mean,
No,
maybe
of course not.
New York and Massachusetts and California and a couple of other states could pick up a good bit of that slack, but it would require moving budgets around from things that need to get done in those states, and that's just no.
We need to get through the midterm so we can change all this,
Yes.
because these changes are not good for you folks. I don't know
No.
if people are listening who are not, the low-hanging fruit, but these changes are not good for you. Okay? I don't care what your political orientation is. These are not good. We have known for years that what we do is good. What our public health policy is good for you. There was never any question about it. And now all of a sudden we're questioning it.
Public
for no
health
good
should be a
reason.
bipartisan issue.
A nonpartisan issue.
A nonpartisan issue, yes.
Yep.
But remember, when you mix science and politics,
you get
politics.
Yes.
And public health is by nature a mashup of
And-
science and politics.
Speaking of nonpartisan, death is nonpartisan, and we have another death in our community. This is a letter from Anthony Doma. Hey, TWiV, it's sad I share that Dr. Dennis O'Callaghan passed. He was chair of the microbiology and immunology department at LSU Health in Shreveport, previous president of ASV. I really wanted to introduce Vincent and Dennis when I scheduled him to come visit on campus when I was there, but that pesky pandemic got in the way. Yeah, never made it. DOC was an icon of virology, receiving the highest award given at LSU. Boyd Professor established a center of molecular and tumor virology and taught thousands of scientists and medical doctors. He instilled hard work in his mentees and was always joking around with some zinger one-liners. Every time he saw me, he asked if I was worried my wife would ever get her vision fixed. Ha, 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 ha. You get it?
Yep.
Yeah, I get it. Took me a while, but I get it.
He secured $40 million of external funding for virology research, with his personal work mostly focusing on alpha herpes virus, gene expression, and viral pathogenesis. And he was the first person to give me a shot at science/virology. Below is more about his life and career. Gives a couple of links. He will be missed. So Dennis O'Callaghan had a southern accent. He always used to call me Racchiolio. What's with that Racchiolio? He actually tried to recruit me years ago. Yeah. I didn't want to go to Louisiana.
Dennis is a good guy.
Yeah.
He's a good guy and really dedicated. If you read this stuff, he was 1,000% science and education. Good for him.
Yeah.
No,
quite a
he
loss.
supported people. He
Yeah.
supported many people who
Yeah.
couldn't get support anywhere else. He believed in them. At least give them a chance to do something. If you don't have a chance, you can't do anything, right?
You can read that in this letter. He was the first person to give me a
Yeah.
shot at science/virology.
Yeah.
And the Wikipedia page you link to, and also the LSU page that you link to, are very informative about him. He was also an avid sportsman. A couple of pictures in there, duck hunting in Canada. Apparently went to Alaska just for the halibut.
Well, he's joining a number of luminaries who have recently gone up there as well. So enjoy. You have your journal clubs. All right, on to the literature. We have a research article in the journal called Neurology. Risk of Alzheimer, dementia after high dose versus standard dose influenza vaccination.
And first author is Avram Samuel Bookbinder and Yaobin Ling, co-first authors. Senior author is Paul Schultz, and the team is from UT Health in Houston and Massachusetts General Hospital in Boston.
This is following an arc where we have done other papers showing links between herpes viruses and dementia, links between shingles vaccine and dementia, reduction of dementia, and now influenza vaccine. So
actually, one of the questions that we discussed on the shingles vaccine arc was, is this specific to the shingles vaccine that you're reducing the risk of dementia? Or could you just go get your flu shot and reduce the risk of dementia? And this paper is addressing exactly that question.
So before we talk about it, so Alzheimer's is one of a couple of different kinds of dementia. It's the most common cause. It accounts for about 60 to 80% of all cases. It's a progressive, irreversible neurodegenerative disorder. It destroys memory. It destroys mental function by gradually destroying neurons. And it starts decades before symptoms appear. So you start to have accumulation of these two proteins, amyloid and tau. They misfold and accumulate in the brain. It may take 15 to 20 years before you see any symptoms. You initially have mild cognitive impairment, right? You can't remember things. And then you have memory gaps. You get lost a lot. You can't find the right words. And then you get increasingly confused, personality changes, and then eventually you have to have full-time care because you can't take care of yourself. There are currently almost 7 million Americans living with Alzheimer's, 50 million worldwide. And in the U.S., it's projected to go to 13 million in 2050. And 1 in 3 seniors die with dementia, I should say. Okay. So this is a serious business.
Very, very serious problem. And it is difficult to definitively diagnose.
I was gonna ask.
So certainly in the late stages, you can say, oh, yeah, that person has dementia. Well, is that Alzheimer's dementia? And for a long, long time, the only way to definitively diagnose it was to look at the brain, slice the brain and look at it. Obviously, you can only do this post-mortem. And that is still the gold standard of diagnosis. Okay, we know this person had Alzheimer's disease because they died and we looked at their brain. There is also now the ability to do a PET scan, positron emission tomography, which, unlike a CAT scan or an MRI, this is an unusual and expensive test that is not generally covered by insurance. So it is uncommonly done, but it can reveal the structural changes in the brain without having to cut the person open. So we do have some data. A lot of what we've learned in recent years comes from these brain imaging techniques, where we can track the course of the disease while people are still alive, rather than waiting until they've died and then seeing the final result of it.
Yeah, that would be useful for one day when we have some treatments. You could start.
And there are a couple of treatments. There's one that's somewhat controversial that's gotten onto the market that is an amyloid targeting treatment. What's controversial about it is it's not entirely clear that that's all that useful, and it is very, very expensive. But it is a treatment. It has some efficacy in some subset of patients at least. And it's possible that if you could diagnose this condition earlier, you might be able to prevent the development. And one of the big issues is that as these plaques and tangles develop in the brain, it's not clear how much of that you're going to be able to reverse. So we'd really like to know at the mild cognitive impairment stage, which can be an early sign of Alzheimer's, or it can just be, your brain is aging. If we could sort out those patients at that stage and say, oh, this person is actually developing Alzheimer's disease, treat then, maybe we could prevent onset.
Do we know whether the plaque and the tangles are really primary to this, or if they're downstream from some other primary cause?
That is right. So there is a controversy, has been for many, many years, a controversy in the field, not only about which way the train runs, but also which of these two things is more important. Amyloid is kind of the defining feature, but the tau tangles are also an important feature, and there's accumulating evidence that it may be that tau is the lead and amyloid polymerization follows. So that again, this is a difficult disease to study. There's also a phenomenon, this is a bit of a digression, but people with Down syndrome, which is caused by a trisomy, trisomy 21. Chromosome 21 has the Alzheimer's precursor protein gene on it. And people with Down syndrome produce excessive amounts of Alzheimer's precursor protein, which causes them to develop early onset Alzheimer's disease. But theirs is different because it's actually the amyloid that leads anyway. That's a little digression.
So besides Alzheimer's, we have Parkinson's, another dementia. We have Lewy body, which Robin Williams died of, and I don't know what any other. You know any others, Alan?
There's a whole panoply of them there.
As you'll see, the diagnosis is a little difficult, and that turns out to affect this kind of study as well.
One of the reasons we're digressing so much into Alzheimer's disease is because it's very, very relevant to the difficulties of this study.
Yes. Also, I don't know much about it, so I'm a virologist. But it turns out viruses are clearly involved.
Yeah.
So the first sentence of this article, accessible, low-cost preventive measures are needed to reduce risk of dementia. And that's so true. And one of them may be vaccines, so the shingles vaccine. And it turns out that several antimicrobial vaccines have been linked with reduced dementia. And these authors have previously shown that influenza vaccines are associated with a 40% lower risk of dementia over a 4-year follow-up. That's pretty good.
Yeah.
Right? I mean, I get a flu shot every year, but after this paper, I'm going to try and find a high-dose flu shot because it's even better. And this is over a 4-year follow-up. It could be even better longer, right?
Yeah.
So the idea is that these vaccines, they decrease severe illness and they decrease systemic inflammation, and therefore they decrease neuroinflammation and neurodegeneration. That's the viral hypothesis. But it could also be non-microbial mechanisms of various sorts, like inflammaging is impaired and immunosenescence and so forth. So it doesn't have to do with the microbe. We really don't know. What we know is that the vaccines seem to have an effect. And they talk about the dementia risk reduction after zoster vaccination. And now the thing that's key here is it's stronger in women than in men. And you're going to see that it's the same with
the effect is stronger,
Yeah.
I was particularly interested to see that effect reproduced.
Yes.
Yeah, it's quite interesting. So anyway, the hypothesis here is the question here is whether the dose influences the effect of influenza vaccination on AD, because we do have normal dose and high dose flu vaccines. And I wanted to tell you a little bit about that because maybe you don't know about it. The regular dose flu vaccine has 15 micrograms of hemagglutinin. The high dose has 4 times higher. It's 60 micrograms. And that's only for 65 and up, the high dose. I can never get it. It's always sold out when I go to get them.
Right. So as you get older, your immune system doesn't hear as well, and you have to shout at it.
That's right. Seroconversion is about 25% higher HAI, hemagglutination inhibition titers, with the high dose versus the standard. It's 24% more efficacious in preventing severe disease than the regular. And it's great. Again, it's much better against hospitalization, preventing hospitalization and mortality. So but you get more injection site pain, it's higher cost. But the CDC ACIP, the old ACIP, had recommended it for 65 and over without question. So
with question, but they had their questions answered.
Yes.
And just to clarify or make sure people understand it, this is a subunit vaccine. There's no virus in it. Okay, so this is the virus attachment protein, basically, much the same as the coronavirus vaccine that has as its active component just spike and no real virus in it.
So this is called Fluzone. What's the other name for it? Right? There's regular and high dose. Let's see how it's made. How is it made?
I'm not sure. Some of these are baculovirus produced.
Yes, I think
but I don't know that that's this one.
Now, Fluzone is inactivated egg grown. Yeah.
Yeah.
But it's, as you say, it's mainly HA protein. All right. So the question they're asking here is, among people 65 years and older, what's the effect of vaccination with a high dose versus standard dose at 3 years, at the 3-year risk of Alzheimer's disease? All right.
And this is a neat way to ask the question, because if you're comparing vaccinated to unvaccinated with the flu shot, you introduce all these biases of people who are more likely to get their flu shot, are more likely to live healthier lives, and have better socioeconomic status, yada yada yada. But if you compare low dose to high dose, these are all people who got their flu shot, and then you're comparing the dosage to see what happened.
And they may, referring to Vincent's thing, they may not have even had a choice.
Maybe they wanted the high dose. Yeah.
Yeah.
Mm. So what they do is they look at a database. It's called the IQVIA Pharmetrics Plus for academics, a U.S. commercial claims database with inpatient, outpatient, and pharmacy claims from all 50 states. And they are able to access the data from January 2014 through September 2022. And what they do here is interesting and is a learning moment here. They have two kinds of analyses. They have a per protocol and intention to treat. Okay. And so per protocol means the people who actually got vaccinated, because in this database, there are a lot of people who could have, but they didn't. Intention to treat, they look at everyone because that's more reflective of the population, right? The population of the U.S., not everyone's going to get a flu vaccine. So you want to know what the overall. But the thing is, in the ITT, they didn't remove people who got subsequent flu vaccinations, which would mess up their protocol. So they focused on the per protocol analysis. But anyway, this is the difference between those two. I think that's worth knowing.
And just a couple of other things to point out about the study design. They had data from January 2014 through September 2022, but they cut off the data July 31st, 2019, so that they wouldn't have the rather large confounding factor that showed up in 2020.
Yes, right. And so, how do they define?
And the other thing, are you going to talk about the target trial emulation?
No, you can talk about it.
Yeah. So they essentially they take this huge data set, and I forget the numbers. They're like
it's a couple of 100,000 people.
Yeah, it's a couple 100,000 people.
Well, it actually starts at 28 million, and then
they winnow it down whether their actual population they're studying. So they then arrange the data in a way that they're essentially going to simulate a clinical trial, as if these people had been randomized into low dose, high dose, no dose groups. And you can do this statistically when you have this large a sample. So it's a synthetic trial because you're not actually conducting the trial, but you're doing it from the data.
And they can see in the records if people have gotten low dose or high dose, and they just segregate them into the two groups. And then how do they define Alzheimer's disease? So they look at the records, the ICD records, right? And they see either Alzheimer's, senile dementia, dementia of unspecified cause, or a claim for a drug used to treat dementia, right? Donepezil, galantamine, rivastigmine, or memantine. So that's, and as you will see, the results get fuzzy when you do that. The best thing is to say Alzheimer's on the medical record, and then you get an effect of the vaccine. And when you do all the other stuff, it's not as good.
Now,
and they talk about
right, and they talk about they talk extensively about the limitations of this approach.
Yeah.
But one of them is people getting diagnosed with Alzheimer's disease. The ICD, the International Classification of Diseases, this is what the insurance companies use. They assign a code to each disease. Right. And so people who've gotten a code, a diagnosis saying Alzheimer's disease, they do point out in the discussion that is sometimes, a diagnosis that's done based on symptoms. And as we discussed, it's usually a diagnosis based on symptoms. As we discussed, that's kind of wobbly. You don't have the PET scan, you don't have the autopsy. So there's some wiggle in the data there.
Yeah. Oh, so they did secondary analyses. So the first is Alzheimer's. In the secondary, they looked at the sex effect, as we mentioned. They also excluded people who were vaccinated in the past 2 years, right? Because they say mimicking a 2-year influenza vaccine washout. They replaced AD with incident MCI, mild cognitive impairment, as the outcome, and treated Alzheimer's as a competing risk. Fourth, because the flu vaccines recommended annually, they assessed AD risk after seasonal treatment with repeated vaccines, both high and regular dose over 3 years. And then they compared adjuvanted versus non-adjuvanted influenza virus. And then they had done a previous study with regular dose influenza vaccine. They wanted to see if this would replicate their findings, right? That's a nice control for that. So, so they start with 28 million people, and they whittle it down to 185,000 high dose, and that's actually 120,000 unique persons, and then 54,000 standard dose, which is 44,000 unique persons. And they divide them into ITT and PP groups.
They also in this trial design, this synthesized trial design, they corrected for something, and this caught my eye, and I had to look it up: immortal time bias, which is not people turning into vampires. It's really, I would call it plot armor. So when you follow participants from enrollment through the start of a treatment and then to some outcome, anybody who enrolls who would have gone to the treatment group but died or fell out of the study or developed some, contraindication or whatever, you wouldn't include them in the treatment group, right? So effectively, the people in the treatment group have this period of immortality because they had to have survived to the treatment. Your control group, if you don't have that same plot armor on them, you skew your results. So they corrected for that as well. They've done a very good job of tracking all the potential confounders here.
This discussion highlights that, for me, one of the real messages of this paper is to do a study like this is really complicated.
Yes.
And the variables are just all over the map, and controlling for everything is really difficult.
But it can be done.
Can be done.
If you have a good enough team.
And this is the team that should look at the CDC data that Bhattacharya doesn't have.
Well, these are the kinds of people who review those papers for MMWR.
Yes.
Yeah.
Okay, so compared with standard influenza vaccine, the cumulative risk of AD was significantly lower after high dose from months 1 to 25 in the per protocol design and 1 to 28 in the ITT design. And the maximum statistically significant risk difference was 0.0054. But the more interesting number is the number needed to treat. So you have to treat 185 people with the high dose to reduce the risk in one person of Alzheimer's, and then the other is 270 people. So actually, the PP design is 185 to treat and 270 in the ITT. So the ITT is the overall population, including people who don't get the vaccine. So it gets diluted a bit. Somewhere, and I don't know where I found it, the risk reduction is about 22%. Okay, it's not easy to find that number here, but because this risk difference, 0.0054, it's hard. I don't know what that means, frankly. I mean, that's a difference between the two treatments, but it's, I like risk reduction better.
Yeah.
And so that's the bottom line. There is a difference between regular and high dose. And that's cool because it's like a dose response curve, right?
Yeah.
So, which normally you can't do in these kinds of studies, but it does give you more confidence that the effect is due to the vaccine, right? And then women had lower risk compared to men. And the results, in fact, the PP results per protocol were not statistically significant, although the ITT results were, but still lower than women.
Right. So women benefited more than men from the vaccine and in prevention of Alzheimer's disease. Men still benefit in prevention of going to the hospital for flu, though.
And this is interesting. No difference in the adjuvant versus non-adjuvanted.
Yes.
It's kind of interesting. And just flu vaccination versus no vaccination, they reproduced their earlier finding that they had already published. So that's cool. And that's using a different database. So that's more support of this idea. It's very cool. And there's a table that reports all this. I think this is open access. So
yes, it is.
The tables, does the primary goal. It tells you, and so this is interesting. It's the primary goal. There's the intention to treat group, protective from months 1 to 28, no difference from months 29 to 35. So it seems to fade. And so that suggests that you should probably get it every year.
You should get some vaccine every year, and your flu shot's as good as any. Yeah.
And then the per protocol, it's protective also from months 1 to 25 and nothing from 25 to 35.
There was a sentence in here that I didn't understand that maybe you can help me with. It says, this is under sensitivity analysis. When the ICD codes, those are the codes that specify exactly what
the
disease
diagnosis
you're talking about. Yeah. For the unspecified and senile dementia were removed from the case identification definition. The number of incident AD cases in each of the high dose and standard dose groups was approximately 80% lower than in the primary analysis. I don't understand the comparisons being made there. And there were no significant differences between the high dose and low dose. Can you clarify that for me? This is
so basically, I don't know. Yeah, when they take out the more less specific, right? More less rigorous. So unspecified and senile dementia.
Right.
When they remove that, then all you have left are ones that say AD. So I don't know why there would be no significant differences. It doesn't make any sense to me. I would think the other, if you would go higher because you're now looking only at a definitive
and
diagnosis.
The primary analysis is the analysis with those ICD codes in there.
Yes.
Okay.
With everything, yeah. But also, if you
one thing I should have gone to the e-tables that in the figure that the supplemental figure they cite here to see what the numbers were. But it may be that when you take out those diagnoses and only include the definitive, well, not definitive, but the ones that said Alzheimer's disease, you may have shrunk the statistical sample too much.
Significantly, yeah.
Yeah, they say there was no significant difference, but there still was a difference, but it doesn't hit statistical significance. Yeah.
The other thing that interested me was this last bit in the results, because, there's a lot of stuff in here that was new to me. It says, a classification of evidence. This study provides class 2 evidence that treatment with high dose versus standard dose is associated with decreased incident dementia in individuals greater than 65 years of age captured in the U.S. healthcare claims database. So class 2 evidence, I had to look that up.
It's observational, right?
Yeah, and realize that there are specific types of evidence for these sorts of studies. And class 2 evidence, one of the things I got out of it is that it's not absolutely certain definitive. There are some, because of the way the study is done, there are complicating factors, which they talk about all through here. And, class 4 evidence is like, nah. And class 1 evidence is, yeah.
Randomized
solid.
control
We
trial.
got this right in. This is for sure.
Trial. Yeah. So observational studies are always like that. You never know. But if you do enough of them with enough people, then you get confidence. That, like if you do 20 studies looking at connection between autism and vaccines, eventually you say, we don't see any connection in any of these studies. So,
like so many other scientific things, reproducibility is what really counts.
Yeah.
Also, when they define Alzheimer's by medication only, there's no difference in the treatment, right? And
because probably people are confused, so they take drugs, and who knows why they were confused.
Right, and when that's going to be an even smaller population.
Yeah, true.
Yeah, it was 60% or something like. They have 2 graphs which are nice: the cumulative incidence of AD with high and standard, and the 2 populations, the intention to treat, and the per protocol. And you can see it's colored, so you can see the reduction in the incidence with the high dose versus the standard dose with time. Right, that's a really nice graph.
Yeah.
And it's always AD. It just keeps reminding me of you, Alan,
Yes.
Class 2 evidence. So there you go. This is, so even though the effect is stronger in women, both men and women do exhibit lower AD risk with the high versus low. It's just that the duration and strength of it is greater among women, right?
Yeah, and
they do say that the duration of reduction may depend on repeated vaccination at least every 2 years, right?
In this, they refer to a herpes study that says that the subunit vaccine, Shingrix, is better than the live attenuated vaccine. We didn't do that paper, right? We did the live
I don't
attenuated
remember that
guy.
one.
And I remember asking that question of ourselves, okay? But that's very interesting. And in the case of the herpes, that's really important because of the latency factor and all that kind of other stuff.
And as somebody who got Shingrix and not the live attenuated, I was very interested to see that.
Yeah, it's good. So you get your Shingrix, you get a flu shot every year, and who knows, maybe COVID vaccines are also reducing.
Absolutely.
This is a recent thing, right? So we're not going to have as long of, but that'll be done one day, and it could be that they're all dementia vaccines, right?
Yeah, I mean, this really supports the idea that we hinted at with the herpes virus papers. Is zoster causing the dementia? Is it, because you're preventing zoster, or is it just because you're getting a vaccine? And this really suggests it's because you're getting a vaccine, at least, some significant part of the effect. And that makes, that's a perfectly reasonable hypothesis, knowing what we know now about inflammaging and what the immune system is doing during aging and how that can be involved in processes like Alzheimer's disease. So you're goosing the immune system, and it's, maybe inflaming the brain less.
My only problem with that is that to me, my immune system is under constant attack. It's being insulted all the time. So it suggests that there's something different about vaccination than just walking around in the world.
Your immune system is constantly dealing with low-level threats, but a vaccination presents a higher level threat, an actual. If you get a flu shot, certainly if you get a COVID-19 shot or Shingrix, you feel the effects. After I got my Shingrix vaccine, I knew there was something going on with my immune system, whereas walking around just inhaling the air doesn't do that to me.
Good point.
So a couple of limitations are worth mentioning. One is that the short follow-up for 3 years. And they say this because this is a long incubation period, so we need to do studies with a longer follow-up. And also the database doesn't have information on socioeconomic lifestyle data, right? Income, education, all that stuff, which they say is a common issue among administrative databases. And so this is the thing that Alan mentioned earlier, right? The healthy lifestyle effect could be at practice here. But, they say we need to know the mechanism so that we could more specifically hone in on how to prevent. But meanwhile, I'm going to get a flu shot every year, and we don't need more than two shingle shots, so that's done.
Yeah.
And I don't know why, in the light of this data, you wouldn't do it.
Yeah.
Because dementia sucks, folks. It sucks. If you know anyone with it, it sucks to see a fully functioning human. Well, not all humans, even at 20, are fully functioning.
Well,
but you know what? Because they're, it's political commentary. But it's just horrible to see them just be unable to do anything, and it shouldn't be.
And you know what else sucks is going to the hospital with flu. So,
yes, it sucks too.
This is not just, this will slightly reduce my chance of dementia. This is your flu shot. You should get it anyway.
Yeah. All right. So now we will do a cell paper. It's called Deltaviruses Spread Through a Viral Trojan Horse.
The first author on this is Joe McKellar. The senior author is Karim Majoub. The team is from University of Montpellier, CNRS Montpellier, France, INSERM in Tours, France, Université Grenoble Alpes, CEA CNRS in Grenoble. Grenoble or Grenoble?
Grenoble.
Grenoble, sorry. Also University of Helsinki and University of Zurich.
So I heard this story last summer at ASV in Montreal, and I went up to Karim. I said, this is beautiful. I want to do it on TWiV. He said, well, I'll let you know when it comes out. And he just emailed me a couple of weeks ago, but I had already seen it come up, and it got some press. So these viruses, these are viruses that need a helper virus in order to replicate. They call them satellite viruses, but in the virology world, they're viruses like anything else. They're just dependent on a helper virus. Viruses are dependent on host cells, and we don't call them satellite viruses. So anyway, they're just viruses. And examples are adeno-associated virus, which we talked about recently because the structure of the rep protein was determined. They need either an adenovirus or a herpes virus helper. And then hepatitis delta virus, which needs hepatitis B virus to provide the glycoproteins to package the genome. And it's thought that if you are co-infected with both delta and hepatitis B virus, you get worse hepatitis. Okay. Now, for a long time, these two were the only known human helper-dependent viruses. And then actually this paper we did on TWiV a number of years ago, they found hepatitis delta-like, HDV-like sequences in a lot of other animal hosts, rodents, bats, birds, snakes, frogs, insects. And so there's a big family of these circular RNA viruses that are throughout the animal kingdom. They're now called Colmeoviridae. But for the other
Here's my number, Colmeoviridae.
Colmeoviridae. They don't know what the helper is, right? They just found the delta genome, and we don't know. There's no Hep B or no any other virus that known to help these viruses.
So how are these other guys getting around?
Right. Now, there's a snake delta virus, which was found in boa constrictors that are infected with Reptarina viruses. And those are the helpers for that. But that's the only one they know the helper. The other ones, they don't know what the helper might be. So they were interested in looking at that in this paper. And they end up finding that these delta viruses can get packaged inside of the helper virus particle. So in addition to stealing the glycoproteins, can actually take a ride inside. So that's a Trojan horse, right? You remember that story? The troops hid in a horse that was given as a gift.
The Greeks hid in the horse and got presented it as a gift, supposedly, and then it was brought into the fortification and they
Mm.
slipped
Boom.
out in the night. And
So
yeah.
that's the same idea. So Trojan is perfect name for that, right?
and another thing that this paper finds is that the Delta viruses are apparently not that picky about which other virus they ride in. So
Yeah.
it's not just, oh, they can only ride with this. It's they may actually be able to hitch a ride with a few different viruses.
Yeah. Well, as you see at the end, they call for more looking in people because
Yes.
we assume that Delta is the only one, but maybe we haven't looked enough, right?
Yeah.
So they start with rodent Delta virus, okay? It's from a Tome's spiny rat. This can replicate in a lot of animal cells. It's packaged by the VSV glycoprotein. So the Delta is the Hep B. So this rodent Delta virus is the VSV. And so they do some experiments where they superinfect a mouse fibroblast cell line, which is persistently infected with this RDEV, rodent Delta virus. And they infect it with VSV, and they look at the virus production. And they see release of Delta antigen into the supernatants of these cells. They do PCR, they can see a 5-fold increase in genome, the Delta genome copies, right? And these in supernatants compared with those with no superinfection. But there is some residual rodent Delta virus production in the absence of VSV. Well, VSV stimulates it massively, but there is a little bit already. So they want to look at what the secreted particles are. So they take supernatants from non-VSV infected cells, and they can see these spherical particles that are not present in the wild type cells, the NIH3T3 cells. So they're suggesting that there's low level secretion of these rodent Delta viruses from these cells. And if you superinfect with VSV, you see the bullet-shaped particles of VSV and also spherical particles, which are like the rodent Delta virus. Then they keep looking and they find a third class of particles of Delta viruses that are physically associated with VSV. They have nice pictures of this here. Lovely bullet shape. These little tiny virions, spherical virions that are associated with them. They're about they're 35 nanometers, a little smaller than the free Delta virions that come out of the cells.
I love microscopy papers because you can look at the figures and just, oh, that's what's going on.
You can see it, yes. Nothing like a photograph, right?
Yeah.
And some of these VSV have more than one particles associated with them. They also see the same VSV-associated particles in cells where they put the woodchuck Delta virus in. So it happens with more than one. Maybe it's a general feature of Delta viruses. Okay, now
so
these
VSV
are all...
could chuck woodchuck virus.
They could. But, to do EMs, you have to fix the samples, and they're worried about fixation artifacts. And so they use bioatomic force microscopy where you don't need to fix the stuff and you can look at it and they see the same things, right? They can see these VSV decorated with multiple particles. And so this happens without any artifact of fixation. And so they're thinking that maybe there are some Delta viruses within these VSV particles. You can see some of them have a little bump. The VSV, there's a little bump, which suggests there might be a particle in there. So they wanted to look at that. So normally the VSV bullet is very smooth, but now and then you see this bump, and they're thinking maybe there's Delta viruses in there, which is it's an unusual idea because nobody has seen that, right?
Right.
I mean, there are some giant viruses that apparently packaged these helper dependent viruses inside of them, but they're all screwed up. They're not even infectious anymore. You can find pictures of that in the literature.
I want to clarify a couple of things before we go, for myself, among other things, before we go too much further. You said so that this rat Delta virus
right.
is said coated with VSVG. Now, that's an experimental condition, right?
Yeah.
Because
Sure,
it's not
sure.
ordinarily transmitted
Right.
that way.
That's experimental, yes.
That they've set it up that way so that they can infect cells with it. Also said that you have cells that are persistently infected with this thing. I don't quite understand what that means. Like a little bit of replication of the RNA, but not necessarily a lot of packaging and transmission.
Yes. Yeah. So you could put DNA, I guess, in encoding this delta, and it will be transcribed into RNA, and that will replicate.
That will replicate.
I believe it's replicated by RNA pol 2.
Right. Yes.
RNA pol 2. Yeah.
Yeah.
And so, in the absence of VSV, you get some particles released, and they're smaller than the ones you would see with VSV, because when you add the glycoprotein of VSV, then it gives it a bigger dimension.
And I'm reminding myself here, the sort of commonality of these Delta viruses is that they're small, circular, self-complementary RNAs that encode a single protein that's required for the replication, but doesn't serve to encapsulate it. So they need to rip a capsid protein off from somebody else in order to actually be transmitted, which is what makes this particularly relevant, because they're apparently hitchhiking. The other thing, and I'm thinking all the way through this paper, that they wind up at least approaching addressing at the end, is that, okay, this is a rat Delta virus on cultured mouse cells, all right, that have been in culture forever, hitchhiking on VSV, which is a cow virus, all right, that is, at least originally, that's a common laboratory tool. So I'm not knocking it. They're setting up the system, okay, with stuff that they can experimentally do. But keep in mind that that's the situation so far. It does not reflect what's going on in nature.
Yeah.
Right. Obviously, they have to look. At the end of this paper, they say we need to look.
Absolutely, yeah.
But, we do know that in humans, Hep B can package Delta in the Hep B envelope, right? But that's different. We've never seen the Delta inside of hepatitis B virions.
I don't know. Have we ever looked?
No, I don't think we looked that way. We just, nobody looked. All right, so they want to try and understand these VSV-associated particles. So they're using antibodies now to stain virus particles by super resolution microscopy, so you could get a really fine resolution of the staining. And they take these NIH 3T3 cells that are making the RDEV without VSV, and you can see the virions. They have pictures of those here. And when you superinfect with VSV, you get the usual elongated VSV particles, smaller free RDEV particles, and a subset of the VSV stain with antibody to the Delta virus on the periphery of the virions, right? So that's telling you that inside the virion, the VSV virion, there's some Delta virus.
So this is pretty amazing. I've seen all the different microscopy tools that they use here. By the way, the atomic force microscopy is you just see surface features. There's a little lever that is dragged across the surface that has a laser shining on it, and every time it hits a bump, it deflects the laser, and that's computed into surface features. This is optical
I think of it as
microscopy.
Braille microscopy.
Yeah, Braille microscopy. That's good. This super resolution microscopy, it's optical microscopy, right?
Yep.
Otherwise, you wouldn't be doing essentially immunofluorescence with it. But I don't know methodologically how this works, but it is at a super resolution. So we're seeing individual virus particles and basically subvirus particles. It's amazing.
Yeah, there's a so super resolution microscopy is it's one of the honest to gosh jaw-dropping breakthroughs of the past, I don't know, 20, 30 years this has been going on. But the short version is it uses some physics and optical tricks to go beyond the diffraction limit. Normally, you're limited in the size with traditional light microscope. You can see down to a certain size, but that's a function of the wavelength of light.
Right.
And you can get a little bit below that by using shorter wavelengths of light, but there are limits to that. Super resolution microscopy, as I say, uses optical tricks to go further. It's still optical microscopy, but it is it's on another level.
Cool.
And this was the people who developed this got a Nobel Prize
Yeah.
in 2014. This is a big deal, right? So it's all about breaking the resolution limit of.
And we aren't done
light
yet. We got more
microscopy.
microscopy for you.
Yeah. Oh, yeah.
Okay, so, hold on, I just hid you guys. I don't want to hide you. Okay, so we're looking at these cells by microscopy. They also use a different kind of approach. They use immunogold labeling followed by electron microscopy. So you can put small gold particles on the antibodies, and when they bind the antigen, then you can look at them under EM, and you can see the gold and they get the same results. They can see the particles accumulating in the VSV virions. So basically, the virions, the VSV-associated structures, the little particles that they found earlier are indeed Delta viruses. They confirmed it by this super resolution and by the immunogold. All right, so this is all in NIH3T3 cells. So let's try a bunch of different cell lines. So they try human embryo 293 cells that are producing rodent delta, which are producing Delta virus. This seems to be a different one, MMDev. And they can show that putting VSV in them gives you particles attached to the VSV. They also tried HUH5, which is a human liver cell line, 7.5, and BHK cells, baby hamster kidney cells. They try primary cells, primary mouse fibroblasts, or human brain tissue explants. Wow, where do they get that from? Surgeries, I guess. Primary mouse ear skin fibroblasts. Organotypic cultures of post-mortem adult human brain explants. And they find in all of them, they can find VSV-mediated production of these Delta viruses, and also in lines from different organs like liver and kidney. So it's not just one, and different species, mouse, human, and hamster. So it's so far good. It's good when it's not an artifact of one cell line.
Yeah.
Right? All right. So what's going on here? What's really happening? So they have this is cool. They have a VSV that is deleted for the gene encoding the spike protein, and it replicates fine. It makes virus particles that they call bald because they don't have VSV, and I object to that name. Okay. I don't think that's right, but okay. And so then they asked, do you need the glycoprotein to drive these associations with the Delta viruses? And in fact, you can see these little things independent of the glycoprotein of VSV. So they don't need, these Delta viruses do not need the glycoprotein to form. If you just mix Delta particles with VSV, they don't form either. And so what it means with no G, independent of G, means you don't need membrane fusion to happen. So it probably happens inside the cell as the virus buds out, right? It's capturing the Delta virions inside of it. So they say, now we want to find out if these deltas are really within the VSV particles. So for this, they're going to do cryo-EM, right? Where they freeze the samples in aqueous medium and take pictures, and then use computational methods to assemble a three-dimensional image of them. And this,
because they felt like they hadn't done enough microscopy.
This shows that the Delta particles are indeed within the VSV virion, and it's a single virus particle in there. So they say this could benefit the Delta virus. Yeah, good, right? Because then it could get in to a lot of
so this
cells.
These are the most convincing pictures of the lot for me. And so I'm
three.
The particles that are packaged are not. They're just the RNA. Is that right? Or do they have any other? They certainly aren't membraned.
No, they're just RNA, and they
Just RNA. And there must be, and I think they mentioned this, if VSVG isn't involved, the idea is that there's some association with the matrix protein or the nucleocapsid. I'm wondering where the specificity is here. They don't really address that, I guess.
No, that's a good question.
Yeah.
Because it's just sitting under the VSV membrane, right? The RNA. And then presumably that happens during the budding process of VSV, right?
I'm also wondering if there isn't some kind of control you could do. I don't know what it is, with some inert thing that's some other RNA or some other particle. There's ancient lore in the poxvirus world, because poxviruses are big. They have their own version of a tegument, which is lateral bodies. That is something between the core and the membrane that packages this and that from either the virus or the cell. And when people first made poxviruses that could make, during infection, chloramphenicol acetyltransferase, which was a marker for a time, which is a bacterial protein that's irrelevant to virology, and purified particles from that, they saw that poxviruses packaged chloramphenicol acetyltransferase. So we call that the hitchhiker syndrome. So it would be good to find a way to discriminate between just, we'll just package whatever's lying around, and, this is for real. On the other hand, if the Delta virus is whatever happens to be lying around and manages to get from one cell to another by a relatively nonspecific association, who cares? That's okay.
It may not need targeting.
Yeah, good point. This is stream of consciousness here, folks.
So I just want to point out that this experiment with Delta G VSV, right? So you don't get the VSV-associated Delta viruses, but you don't get the free Delta viruses with the VSV-G anymore, right? Because it's gone. So when you infect a Delta virus producing cells with VSV, you get two kinds of Delta. Ones that are in the particle that don't have a separate membrane around them, and then free-formed ones which have a membrane and the VSV-G in them. And then when you take away the VSV-G, you no longer get those free Delta particles, but you do get the RNA inside the VSV particle. Okay. So then they look to try and see when this is happening, and they find that these go in at during the production of VSV particles, and they get inside the particle, and they're basically can be transmitted to other cells. So they call it the Trojan horse spread of this virus particle.
I do have a quibble with that terminology. This is not really that analogy breaks down because the Delta virus is not replacing anything, and the VSV would normally have been making this trip anyway. So the Delta virus didn't create some new shell that looks like VSV. VSV just happened to scoop it up. It's really more like if you need a Greek myth origin story, it's more like Odysseus riding the sheep out of the Cyclops cave. This is a hitchhiking. It's not a Trojan horse. But there we go.
Well, there are also issues too because the Trojan horse was done on purpose, right?
Yes, that's what I was getting.
That's the point. Yeah.
And this is just accidental.
This is really more of a stowaway situation.
Yeah. All right, so it turns out that VSV kills cells too quickly to really be able to study replication of Delta virus, right? So VSV is the infectious cycle is 6 hours, and it kills cell. So whereas the Delta virus takes a few days to replicate. So they want to find another virus that takes longer to replicate so they could watch Delta replication. So they look at herpes simplex virus. It's a big double-stranded DNA virus. It takes a couple of days to do CPE, 2 to 3 days. It's a large virus particle. It's 200 nanometers, and so it could accommodate the Delta particle. So they have their 3T3 cells making the rodent Delta. they infect it with herpes simplex virus with GFP in it, and they see a 2,400-fold increase in extracellular secretion of genomes compared with baseline. And so they actually their electron microscopy didn't work, and cryo-EM didn't work either. So they did antibody staining, um, followed by airy scan microscopy. It's another kind of microscopy. And so you can see if you superinfect with herpes simplex, you get free particles of herpes and free particles of the delta. So presumably the herpes glycoproteins are allowing formation of delta particles, right? And they also have herpes particles that you can stain them with antibodies to delta antigen. You can see it in the particles. So they seem to be incorporated into the simplex particle just like it was in VSV. They can separate the particles now because herpes is so big, right? They can separate the delta particles from herpes either with or without delta. And so they take, and most of the, when they centrifuge these cell extracts, most of the particles are in the supernatant, and the herpes simplexes are in the pellet. So they use the different fractions to inoculate viro cells in the presence of acyclovir, which inhibits simplex replication. They want to monitor delta replication. And what they find is the supernatants containing the free delta particles can't initiate a delta infection. Yet they could when they initiated the infection with the pellet, which has simplex particles with delta virus in it. Okay, so, and they can see this by staining. And so it's interesting. So the delta particles with the herpes glycoproteins can't infect cells. And so they say that then that means that the infectivity depends on the delta being inside of herpes simplex virus. Trojan horse.
Yeah.
Okay. So this is...
Or stowaway.
Or stowaway. And it's not a natural thing. It's an experimental thing, but it just shows you that there are situations where the stowaway is the only way that this virus is going to propagate. So that could be in nature as well, right?
And an interesting aspect of this is VSV and HSV bud in very different ways, right? I mean, VSV comes out of the plasma membrane. Herpes simplex goes into the ER, and yet both of these seem to be able to pick up delta antigen as a hitchhiker.
I actually had to review my herpes virus lore to remember exactly what goes on in developing herpes. And it's really interesting in the context of this because herpes, as I said before, is one of a few viruses that have what they call a tegument. That is, there's an internal capsid with the genome, and there's a membrane surrounding that. And in between those two are other proteins, many of them being viral proteins that are usually packaged there, if you want to be anthropomorphic with they're proteins that have immune modulating effects early during infection that sort of set up the environment. And the way that particle gets made in the case of herpes viruses is that the capsid gets made in the nucleus and buds into the nuclear membrane is two membranes. It buds into the internuclear membrane, so now it's enveloped in the intermembrane space and then fuses with the outer membrane. So now we have the naked particle in the cytoplasm again, and it's there that it acquires the tegument. And then it buds through the Golgi and gets out. So in my mind, it's that stage where it's in the cytoplasm, presumably, where it has the opportunity as it's gathering tegument to pick up the delta particle. I suppose you could say that for the nucleus as well, but I don't know where the delta particles probably hang out in the cytoplasm, right? Because they're
they replicate with Pol II, right?
So that could be the nucleus.
They're in the nucleus. Yeah.
Okay.
So that could happen.
So maybe they pick them up early on. Okay.
Yeah. Yep. So they also try and add herpes glycoproteins to these cells that are producing our delta virus, and it does not work. It doesn't. So they can't. So if you put a VSV glycoprotein alone in these cells, the DNA for it, they will come out. The deltas will come out as particles, but it doesn't work with herpes glycoproteins. It doesn't work with cytomegalovirus glycoproteins, another herpes virus. So they say for herpes simplex is a mandatory reliance on the stealth mechanism.
Yeah.
Okay. But we don't even know in nature if these are happening with herpes. People have to look now, right?
Right.
Which people probably will do.
And despite my objection, we can continue calling it a Trojan horse.
Okay,
mechanism.
I didn't want to.
I don't want. I don't want to inhibit you on that because that is how the authors refer to it throughout.
Yeah,
I'm just saying, if we want to be mythologically correct, that's not entirely.
So then they looked in other cell lines to see if the herpes simplex virus packaging worked, and yes, it's the same thing, basically another cell type. So, but what they say is that. So they say this highlights the tropism and host shifting capacity that this kind of transmission, Trojan horse transmission, enables when you're in herpes virions, because they can go into epithelial cells and neuronal cells, and that's how it can only spread within herpes viruses. So it's going to adopt the tropism of whatever virus is packaging it. All right, so now we have. So they say these associations have not been observed in naturally infected animals, but there is a Delta virus, snake Delta virus, which has been associated with arenaviruses in boa constrictors. So this is a rept. So they used the reptarena virus, which is called the University of Gießen virus 1, and they take boa constrictor kidney cells, oh my gosh, that are making this snake Delta virus, and they infect them with the University of Gießen virus, take the supernatant.
That's an arenavirus, which is a membraned RNA virus where the RNA is in a nucleocapsid inside. Right. Well,
it infects snakes.
It infects snakes.
It's a snake virus. They get the supernatant. They say, we saw modest increase in Delta RNA secretion. Majority of the Delta antigen and Delta RNA are in the supernatant fraction. A minority are pelleting with the Gießen virus. Okay. And about 12% of the particles. So then they look in, is it another cell? No, it's the same cell type. About 12% of the particles made in these cells, these UGV infected cells, which are also making Delta, these particles have an additional density within the virion. They're slightly deformed, and looks like using antibodies, they can show that these are the Delta particles inside the Gießen virus particles. About 20% of them have that. All right. So basically, these are Deltas packaged into Gießen. And when that's the case, you get a 2,000-fold higher infectivity versus the virus particles in the supernatant fraction. And so basically these Trojan horse virions, the Gießen virus with the Delta, snake Delta virus inside them, are more infectious than free Delta particles decorated with Gießen virus glycoproteins. So that's interesting. Because, with Delta, we assume that the free particles are really infectious, and right. That's it. But here, the ones associated with the helper are more infectious as well. And the key here is that this is a natural system, right? It's a snake virus and a snake Delta virus. So I guess you could go out and look in snakes now, but I like snakes. I don't think you should hurt snakes. Snakes are really cool.
Snakes are pretty cool. But you could look in a bunch of other places since these Delta viruses have shown up elsewhere.
Yeah. So basically the finding here is that Delta viruses, animal Delta viruses, can be packaged within helper virus particles, which is something we didn't know.
Yeah.
We thought they just used their glycoproteins, but and they say this may explain why we find Delta genomes in lots of different animals and different tissues, because they can be packaged by different viruses.
And it suggests that these Delta genomes could spill over into more species even than we've seen them in.
Yes. Yes.
So I have a correction/question here. I raised the issue earlier as to whether we were looking at just RNA or a RNA protein particle with these viruses. And it occurred to me they're doing immunostaining in the super resolution
No, yes, of course.
microscopy.
Oh, right,
Yes.
And they use the rat
Delta
delta
antigen.
virus antigen. So there is
Yeah,
some
yes.
association
Of course.
between the delta virus antigen and the RNA.
Yeah, so that normally would package the RNA in like a nucleocapsid,
Right.
and then that would get packaged in an envelope. So that's exactly what is present in these virus particles, not naked RNA. Yeah. So they say it's conceivable that Delta virus has evolved an inefficient yet highly generalist co-packaging strategy, enabling them to associate with a wide range of viral families with diverse assembly and budding mechanisms. So they have different rates of incorporation into these different particles. They're always low, 25%, 33%, 19%. So, but it may work it gives you other advantages, right? It expands
Yep.
your range, and so that may be tolerable. First known example of such a virus contained with an animal virus particle. So they say we should look in humans, right? We should monitor across different human tissues and organs. I mean, there are lots of transcriptomic databases that you could mine for this now. I'm sure people have already done it, though, right? But I would not limit it to Delta. I would search with other human Delta, other Delta viruses, and see if you find them in human tissues, right? In the future, it would be important to show the existence of Trojan horse particles isolated from the wild. Yes, I agree with that. That
Yeah.
would be good. But it's pretty cool.
Yeah.
Never stops.
No, it shouldn't.
Always surprises.
You get surprises when you follow your curiosity, right?
Yep.
Like we say, why are there so many Delta genomes in the world? What could be going on? Let's just start studying it and see what happens. That's what you have to do. You just have to study it. Let's do a couple of emails here. Rich, can you take that first one?
Sure. Charles writes, hello, Twivers. 48 Fahrenheit, 9C, and heading back towards winter in Chapel Hill, North Carolina. John Prine, am I pronouncing that correctly?
Yep.
Prine,
Yeah,
died about 6 years ago, an early victim of COVID-19. His voice wasn't the best. His music style was a mix of bluegrass, country, and stuff I don't know. His lyrics were multi-level and hard to classify. I am going to link to 3 of his songs. The first is unfortunately as relevant today as it was written in 1968, called Your Flag Decal Won't Get You Into Heaven Anymore. Written around the same time and also still relevant is Sam Stone. The last is called Paradise, the unofficial state song of Kentucky. All 3 were on his 1971 debut album. I'm going to add a 4th, much more lighthearted song, Space Monkey. And he lists YouTube links to all those. He says, I hope you enjoy these. I promise they are not as odd as they're made out of meat. Thanks, Charles. Well, Charles, I have to say that I confess that I didn't know who John Prine was. So I looked him up and I listened to all these songs. And man, do you have a convert. This guy is cool. And these songs are great. His lyrics are amazing. There's this sort of black humor and cynicism. They're just dripping with this stuff, but humorous nevertheless. And I wouldn't even call Space Monkey a lighthearted song. To me, that is an animal rights song in disguise. This stuff is brilliant. So thank you.
All right. I'll check it out. I haven't listened. Alan, can you take the next one?
Sure. Anne writes, hello, Professor Racaniello. Heard about this today on a podcast. I wonder if it's a factor against all influenza-like illnesses, including COVID. And I had not looked at this. Dermcidin has antiviral activity and protects against influenza.
So dermcidin is one of these peptides made by
Oh.
bacteria in your skin.
Yeah.
It's pretty cool.
Huh.
And then they used it and they saw that it can inhibit and protect mice against flu. But it's also in your saliva and lots of other places.
Right.
It's interesting. Although peptides are hard therapeutics, right?
Yes.
Yeah.
I'm not sure how much you're going to get out of that, but that's really interesting.
I can take another one since that
Yes,
was very
please.
short. Praneel writes, Hi, TWiV. I am currently a molecular pathology master's student at USC and have fallen in love with virology and immunology through your podcasts, which have provided much-needed respite from my traffic-jammed commute through LA. I am reaching out because I know there has been a lot of criticism, and rightfully so, of the Jay Bhattacharya-directed NIH, especially on the podcast. He and a few other speakers are coming to campus and will field a few questions from the student body. So I was curious to see what questions you all would ask him if you had the opportunity. As a side note, I did a deep dive on the ICTV taxonomy release after it was mentioned on TWiV 1303, since I had never even considered that there are binomial scientific classifications for viruses. As part of the C-Phages program in undergrad, I did some environmental phage discovery work with my classmates and isolated a few different Arthrobacter phages. The program gave near full, without the bounds of reason, agency, or within the bounds of reason, agency over naming phages that students had discovered. And one of my classmates was a huge Nicki Minaj fan, ultimately naming the phage they had isolated colonol barbs, in reference to the title Nicki Minaj's giant fan community has given themselves. I was wondering how this would tie into the official virus nomenclature, and was happy to see it is officially the species name of the virus, Bailey blue virus colonol barbs. I am curious to see how future species will be named as Gen Z and beyond are handed the reins over nomenclature. Thanks for the important science education work y'all do. The podcast has seriously been an inspiration for me and the type of work I want to do in the future, so keep speaking the truth. Sincerely, Praneel.
Yeah, the C-Phages program is great.
Oh, yeah, C-Phages is awesome.
Really like that.
And
so
questions for Jay Bhattacharya, I don't think I would bother.
I don't know where to start.
Yeah.
I would first say, why are you such an asshole? And that's, he's not going to answer that. He's probably
Does
not
your
even gonna let me.
cynicism have any boundaries at all?
I would say vaccines have shown to be an excellent public health measure for, 50 years, 75 years. So why are you not supporting them? You
Centuries.
and your comrades.
Yeah.
And he would say, well, I am supporting them, blah, blah. Right?
Yeah,
he would lie.
He would just evade.
So there's nothing you can ask him that he won't lie about.
I might ask him, how do you sleep at night?
That's a good one. That's a really good one. I would ask him, when are you going to step down?
Yeah.
Because you're totally inappropriate for this position. So basically, none of us can really ask him. Maybe Alan could, because he could be calm.
No, I could not. I would not bother. You're not going to get any kind of a useful answer out of this idiot.
No, he or any of the administration people, they just lie.
Yeah.
Right? Arlo writes, Dear TWiV team, as a prescript and introduction, please cut extraneous info, but I hope you can share my message. Another time I may have apologized for being so passionate lengthily. Concise writing is better received, and I see its value. That said, I refute the idea that we can divorce our humanity from our science. These things have never been mutually exclusive, and I don't think I'm alone in my exhaustion with this idea. I can easily be seen as a snowflake. I am visibly queer, transgender, disabled, and aligned with leftist politics. I'm also early in my hopefully academic career, PhD candidate, less than a year from defending, studying virology in Canada. I've been a TWiV listener since 2018, and while viruses were cool enough to reel me in on their own, continued to enjoy the podcast and learn from you all each week. This is my first time writing in. At this point, it is a moral imperative to speak out, and I thank you for taking it on because your voices are much more easily heard by many than mine. We may not agree on everything, but we can all unite on human health, and I hope that these discussions do not stop until things truly change. I've been wanting to reach out for a while regarding the increase in political discussions and outcry from people who do not like it. First, I want to praise your continued discussion of politics and criticism of poor faith, uninformed decisions by your administration. Your voices are one thing that has helped me to remain hopeful in spite of everything, and I truly appreciate all of your work you are doing for science communication. Something that has troubled me throughout my career so far that I don't think I have heard yet on TWiV is the fact that being apolitical is a privilege. Even in the best of times, nothing we can do can be truly separate from the systems in which we live. While it may be easy for some to ignore current societal issues and political topics, please recognize that scientists are people. Further, some scientists are marginalized people. For many of us, it's impossible to ignore politics. Current administrations are attacking our basic human rights. They do not want many of us to exist, let alone speak. If you can live in the current day without being disappointed and speaking up for human rights and safety, i.e. through supporting evidence-based vaccine schedules, you are likely willfully ignorant and aligning with fascism. Everything is political. The privilege of not seeing that is one to be deconstructed and challenged. The discomfort you may feel in hearing it is not something to push away and continue ignoring. Sit with it, break it down, learn with it. We don't all have to be loudly political or agree on everything, but I hope we can all be human and continue to make space for each other in our complexity. I don't believe we should have to talk about the ins and outs of left versus right, frankly. I wish I didn't have to think about it at all. I'm here for the science, but it's impossible for me to separate myself from politics that are trying to erase me, and I'm tired of being labeled political when I'm simply trying to live my life. Thank you. Arlo is in Canada where it's -3°C with cloudy and scattered snow showers.
Well written, Arlo. Thank you.
Thank you, Arlie.
You're going places.
Yeah.
That's awesome.
It's true. It's all true. Everything is political. Alan, can you, sorry, Rich, the last one. Yeah.
Presley writes, Dear TWiV gang, on Vincent's live stream, I asked if I should get the HPV vaccine, that's human papillomavirus, and I just wanted to write saying I did. I got it a week after asking, and while I have a good impression of vaccines, mainly because of TWiV, my mom was actively telling me not to get it because of various reasons, so I just felt nervous and I didn't know who to ask before I started listening to TWiV. Before I started living to TWiV, I thought some vaccines were okay and others caused autism and diabetes because that's just what I was taught. I actually started listening to TWiV because I was told that COVID was made in a lab and I wanted to learn more about that. Obviously, I soon learned I was wrong and instead learned about so many different viruses and vaccines. And I'm very grateful for your amazing podcast and all the work you put into it. Ever since listening to TWiV, my entire perspective on viruses and vaccines has changed. I love viruses, and I'm definitely going to be a virologist when I grow older. Virology is such a fascinating subject, and I listen to your podcast whenever I can and really enjoy it. Thank you for educating the public and making this podcast. I love listening to TWiV and learning about viruses. I specifically want to work on coronaviruses if I can. I love the history of them. I think their crown surfaces are so cool, and they really stand out to me. Also, they technically got me into virology, and I have a billion more reasons, but I don't want to yap. It's 7C in the middle of England with clear skies. Presley, wonderful. What an
Welcome to the
letter.
team, Presley.
Yeah. I'm glad we infected you. This is awesome.
This is what we want to do, is to give people facts so they can make good decisions.
And keep writing as you move through college and into graduate school.
Yeah,
it's hard.
Absolutely.
It's hard to do. So I'm really glad to see at least one example. All right, time for some picks of the week. Rich, what do you have for us?
Okay, so the latest cool thing going on, one of the latest cool things going on, is the Artemis moon mission. And I have been frustrated for some time trying to find a good graphic of the actual path of the thing. And so the astronomy picture of the day, one of Kathy's favorite resources, came up with something that is an animation of the whole flight path of Artemis 2, and it's really good. It's only a minute and 40 seconds long.
Yeah.
What I love about all this stuff that you don't really think about all the time is that the moon is a moving target. Okay? And the people who compute these paths and figure all this stuff out just boggle the mind. I would add that the other thing that was hard for me to find out was whether the return trip was just a slingshot around the moon or whether there was some sort of powered thing. And it's slingshot.
Yeah.
The moon's
No
gravity
power,
does the whole thing. And the other thing I saw the other day that I need to confirm this independently is that this spacecraft apparently has a weird reentry that is a skip. Okay, so it hits the atmosphere and bounces back a little bit, and that slows it down, and then it does its final descent.
Oh.
I need to confirm that because I always thought that was a essentially fatal maneuver. Okay, that one of the dangers of reentry is that if you don't have the right trajectory, you can actually skip off the atmosphere and out into infinity and beyond. Okay, but this apparently uses that phenomenon as an initial break. But like I said, I'll have to confirm that somewhere. At any rate, go Artemis. We're looking
So
at
I watched this. It looked like it missed.
Well, it does. That's because the moon is moving, right?
Yeah.
It's just got like a fraction of a second.
It goes past the moon, and the moon
Loops back.
The moon's gravity loops it around, which is important because you wouldn't want to be moving at that speed and have to turn around on your own. So the slingshot maneuver is critical, and then comes back. And I also liked when this came up. I have APOD in my feed, so I saw this on the 6th when it came up. I was like, oh, cool. I'm glad Rich picked it. Because another thing I like about this video is they show the magnetosphere, the magnetic field around the Earth, and that the astronauts are going outside of that and then coming back into it, which is important because you need a lot more radiation shielding when you're outside that.
So is it correct? They only went around the moon once, right?
Correct. They just went around the
came right
backside. They didn't even do a complete orbit because they're going too damn fast.
Yeah.
Okay.
Well, if they would have had to hit rockets to slow down, right?
Yes.
And then to leave, they would need to have
They would
another
need to
rocket.
fire again. The other interesting thing is that the original orbit of Earth is really lopsided. Okay. And that's probably part of the computation as well.
Yep.
This all reminds me, my wife and I were on a walk the other day talking about all this stuff. Reminds me, these calculations are fascinating. The woman who did all the calculations for the Apollo mission was a woman named Katherine Johnson. I think there's even a movie about her. I'm looking at her here on Wikipedia. She died in 2020 at the age of 101.
Right.
For you.
So, the equations they use for these calculations are all Newtonian equations because even though they're not correct, they work, as Feynman says, 99% of the time.
As long as you're made
enough.
up of enough atoms and moving at speeds far enough below the speed of light, you don't have to worry about you do have to worry about special relativity if you need really, really, really precise timing.
Yeah.
Which is why GPS satellites have to deal with that.
That's right.
If you just need to make a lap around the moon, you're okay with Newtonian.
That's right.
physics.
You have to correct the GPS. Otherwise, everybody's making the wrong turns. Yeah.
Oh, cool. Katherine Johnson got the Presidential Medal of Freedom in 2015. Good for her.
I don't think Trump has given out any medals, have he?
Don't
I don't even wanna know
go
who he
there
would give
because
them
some of
to.
the medals he has given out just make my skin crawl.
Yeah.
Oh, he has given out some. Okay.
I don't know about. I'm thinking about the last administration. It's just.
Yeah.
Alan, what do you have for us?
I have a pick that's about cherry blossoms, and this is not just to troll Vincent. So
Thank
for
you.
those unfamiliar with the history here, Vincent made a remark about not liking cherry blossoms, and we keep bringing this up. Because who doesn't like cherry blossoms? I mean, this
Only
springtime,
me, I guess.
they're beautiful. We have a similar joke in my household that I was in a bad mood one day when I think Laura said, "Oh, the daffodils are blooming." I was like, "Yes, I hate those," which I don't hate daffodils, but that became like a standing joke. That, oh, Alan hates daffodils. Anyway, so the cherry blossoms are a big deal in Japan. They've had a 1,000-year tradition of celebrating the blooming of the cherry blossoms as a celebration of spring. It's an important tree culturally. And since 1950-something, they have been keeping, 1953, the Japanese Meteorological Agency has recorded the bloom times and intensity of the bloom as it moves north through Japan. This turns out to be now the longest running continuous record of bloom times in any flowering plant that we have. And one of the well-documented phenomena is that they've been blooming earlier and earlier each spring because of climate change. So that's already known, and we know this in bloom times of other things. But this story that I'm linking to the Science News story, but you can also follow the link to the original research publication. They analyzed these data not only for bloom times, but also for the blooming intensity and when the trees reached peak blossoming. So, what percentage of the blossoms on the trees are blooming. And it turns out not only are they blooming later, but often they're not reaching peak bloom. So fewer of the blooms on the trees are coming out, which is really interesting and very disturbing, and turns out to be because cherry trees, like a lot of fruit trees in temperate climates, they need a particular winter cold spell in order to bloom properly in the spring. And if you don't get enough cold in the winter, you don't get a lot of blooms in the spring. And so in addition to the timing changing, the intensity of the bloom has changed because the winters are not as cold. And they're not sufficiently cold, especially in southern Japan, to get peak blooms. So you have these disappointing blooms. And that phenomenon is likely to move north. And I thought that was an interesting finding, and also, potentially very disturbing, because the world depends on a lot of fruit trees, many of which are in temperate climates and have this same sort of phenomenon going on. Here in Massachusetts, when we don't have a very severe winter, we don't have as many apples the next year.
Mm.
So this could have a significant impact on food supplies.
Yep.
All right. So, I think in the back of my mind, this whole climate change thing bothers me as much as anything else, if not more.
Yeah.
Okay?
Yeah.
So
Yeah.
for the record, folks out there, climate change is real. Humans are causing it. There are climate change deniers all over the place, and the only reason for it is because they've got money invested in fossil fuels. And so if climate change is not real, if climate change is real, that means that you can't burn fossil fuels, and that it impacts your short-term financial gain. Wake up. We're poisoning the planet. And if that doesn't do it for you, there's a similar phenomenon going on in Africa where they're having trouble growing cocoa trees. Okay? So climate change is going to restrict our chocolate supply. Right?
It's going to mess up everything.
If that doesn't grab you, you got, wake up. Wake
And
up.
our EPA in this country, which used to mitigate some of these things, is now destroyed.
Yeah. So what do? We're bombing each other instead. Wake up.
Which contributes significantly to emissions, because you got to fly the bombs there, and those jets are not very efficient.
Yep.
Anyway.
I got to rant. I don't rant very often. No.
Yeah.
That's
yeah,
good. Good to hear it. My pick is, I'm trying to learn physics. Okay. So I'm reading books, and this one, I had one last time by Italian guy whose name I forget, but this one is by Brian Greene, The Fabric of the Cosmos, Space-Time and the Texture of Reality. So Brian is a professor of physics at Columbia, and it's a good science communicator. And this is a layperson's book about space-time, relativity, special and general relativity, the origin of the universe, entropy. So what I really enjoy is this discussion of the directionality of time, right? For physics, it doesn't matter forward or back. None of the physics equations care, but for us, we care about time, right? So he tries to dissect that out. Is there an arrow to time? And then he talks about entropy, right? Where systems tend to have more entropy as time goes on. So that gives an arrow to time. And all these things, the origin of the universe are well discussed, but I still have a lot of trouble wrapping my head around, especially quantum entanglement, right?
Yeah.
I think we are limited because we live in a three-dimensional universe that's a fairly macro thing. And a lot of these quantum phenomena, we have no way to think about them. They're
Yeah.
way outside of our experience. And they're cool.
Well, we people who have gone deep into this can mathematically explain them.
Yes, that's the thing. Yes.
And that's great, but conceptually it doesn't.
No.
I mean, the human mind just doesn't work that way. We need math to be able to do it, and then we get these answers like time doesn't have a direction that just doesn't align with our experience of life.
And I'm with you, Vincent. I love the time thing. Just thinking about it.
Yeah, thinking about it is a lot of fun. And you're right. If I think if you were good at math and looked at the equations, you would get it, right?
Right. Get it. You would get it intellectually and mathematically.
Yeah.
But experientially, I don't think we can.
But you think of space, right? The universe is so big, right? And it started with like a pinpoint, which just right there. How the hell can that happen physically, right? And then this whole idea that you're out, there's no directionality. Time is completely gone, because what does time mean when a star is, the light you see is, it coming from a million years ago.
Years old.
So their present is not yours. I don't know. So I think if we went into deep space, people would get screwed up because, of all this stuff. But anyway, I enjoy it, but it's challenging, and I need to read more to understand this whole field.
Well, you got more energy than I do. I would have trouble tackling these, but
I read this book is actually really good. I read it years ago and really enjoyed it.
I should take a course of his, actually, but it's going to be too hard. I don't know. I can't do the equations. I'm sorry. All right, so we have a couple listener picks. One is from Bob. A few weeks ago, my wife and I watched a TV series based on a Stephen King novel called 11/22/1963. Some of us older folks still remember where we were when we heard about President Kennedy being assassinated. The book is centered around a time traveler's attempt to return to stop this from happening. But I picked this because of the amazing story about life in the late 1950s and early 60s U.S. and how much things have changed since then. Stephen King has always an amazing ability to observe and wind his observations into the story. In particular, there is this quote from near the middle of the book in a scene in which there's a discussion about the coming 1964 election, but never underestimate the American bourgeoisie's capacity to embrace fascism under the name of populism. He wrote this sometime in 2009, 2010 timeframe, but it seems so apropos today. At 850 pages, this book never failed to hold my attention, even though I knew the outcome from having seen the series. As always, thanks for continuing to bring real science to the podcast world. It's always a good time, and I learned a lot, Bob. Cool. Oh my gosh, 1964. Wasn't Hubert Humphrey running in 1960? No, that was 68. Who was running in 64? Lyndon Johnson versus... Yeah, it must have been, because that was the end of Kennedy's first term, right? It would have been Johnson versus who? Barry Goldwater, wasn't it? Yeah, It was Barry Goldwater. Anyway, Ann writes, space and rocket fans will enjoy this video. This is like the first 8.5 seconds of the Artemis 2 launch. You'll really slow down. You can see those bloody engines. They're so powerful. But it's a bomb, folks. It's a controlled bomb. I wouldn't get on top of that. I'm not brave enough. And this one from Torsten, great visualization. This feels fragile. How a satellite smashing chain reaction could spiral out of control. Today the space around Earth can no longer be considered empty. More than 30,000 objects are in orbit, and that figure is rising exponentially. So yeah, you got to scroll through it and look at it. Oh, are you scrolling? You're seeing all the things accumulating around Earth. It goes from 1 to a lot. Holy crap.
Yeah.
We're so stupid sometimes.
I was just going to say something like that. We're idiots.
I mean, because of it's really money, like Musk throwing up satellites so he can make frickin' money, right?
Yeah.
A lot of it is.
Anyway, it's cool. It's very cool.
I'm always reminded of, going back to climate change, this political cartoon of a bunch of guys in suits around a table, sitting around a table with a city in the background, and they're up to their chests in water. And the title is something like, politicians debating climate change.
Right. Okay, that'll do it for TWiV 1313. You can find the show notes at microbe.tv/TWiV. You can send us questions or comments to twiv@microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. I enjoy talking with you guys and the opportunity to rant a little bit.
Always. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.